In today's lecture, we will discuss the next topic under regulation of gene expression, that is Lac Operon. In the previous lecture, we discussed about how to regulate a gene. We told that the regulation of gene is not a simple way to control it. So there are many ways. In prokaryotes, there is only one way. If you want to regulate gene expression in prokaryotes, there is a transcriptional initiation itself we need to stop. Whereas in eukaryotes, if we want to regulate the gene expression, we have 4 levels to go. First one is transcriptional level, second one is processing level, third one is transport level, and fourth one is translational level we discussed. And then we told like expression of gene is under metabolic control, is under environmental control, is under physiological control also. All these things, they control it, and the genes or the organism should adapt to the changes in the environment. If not, it cannot survive. That also, what is a necessity also we discussed. And then I told all the billions of cells which are present in our body are perfectly regulated, are perfectly coordinated. So without any mishap, they function normally. If it's a diseased condition, that's a different scenario, right? So some cells will express more, some cells will express less. They'll not code for the protein. Like see, like what is that? Pancreatic cells. If they don't produce the insulin hormone, islets of Langerhans, if they don't produce the insulin hormone, it leads to a hormonal disease called diabetes mellitus, ADH deficiency causes diabetes insipidus, right? So like that, they are less production, means if genes are not able to produce the required amount of insulin or glucagon or vasopressin, like that. So let's not deviate from the topic. Our topic is regulation of gene. So we can explain it in prokaryotes. We can explain it by taking Lac Operon as an example. So let's start. Our topic is 6.8.1. What is that? The Lac Operon. It is called the Lac Operon. So we are in the 6th chapter. We are doing this topic 6.8.1, which is called as Lac Operon. I think I told you, like Benzer is a scientist who coined some names for the genes. Like he used a word called cistron, he used a word called recon, he used a word called muton, right, to define the genes. Cistron is a unit of a gene which codes for a protein. Recon is a unit of a gene which undergoes recombination. Muton is a unit of a gene which undergoes mutation. So what is cistron? Cistron is unit of gene which codes for a protein. Which codes for a protein. Understood, children? Benzer used 3 words to explain gene. He used the words called cistron, recon and muton. Cistron is a unit of a gene which codes for a protein. Recon is a unit of a gene which undergoes a recombination. So, recon is unit of gene which undergoes. It undergoes, what does it undergo? Recombination. It can undergo recombination. Similarly, muton also is a unit of a gene. Muton is also unit of gene which, now you tell me, which undergoes, it is muton, right, which undergoes mutation, Now, similarly, there's a new word, operon. What is that word called? Operon. Who coined the term operon? The term operon was coined by 2 scientists. Their names are Jacob and Monod. So, what are the names of the scientists? We will write separately. Again, Jacob and Monod, right? Jacob and Monod coined the term operon. Now, what is operon? Operon is a unit of a gene which is under regulation. Gene which can be regulated is called as operon. That's what Jacob and Monod told. So, operon means it is a gene which is under regulation. It is a unit of gene which is under regulation. What is it called? It is called operon. Who coined the term operon? The term operon was coined by Jacob and Monod. To explain the concept of operon, they have, in which year? In the year 1961. In the year 1961, like F. Jacob, who is a geneticist. He is a genetics professor with J. Monad. Now, J. Monad is a biochemist. Who is Monad, children? Monad is a biochemist. Now, in the year 1961, Jacob, who is a geneticist, and Monad, who is a biochemist, they proposed a model to explain the regulation of a gene. They proposed Operon model. They proposed a model called Operon model to explain the concept of gene regulation in prokaryotes. They proposed Operon model to explain what they want to explain, to explain the concept of gene regulation. Explain the concept of gene regulation in which organisms? In bacteria, children. Bacteria is a prokaryote. In prokaryotes, so you can understand the concept of regulation of gene by taking Operon as an example. Understood? So then, as I told you here, new terms like cistron, muton, recon, Operon. Let us take two more things also and let us see. So we know what is a structural gene. Let's see what is a structural gene. Sorry. A structural gene means it is the part of the transcriptional unit which codes for a protein. A structural gene means which gets translated. So, what is structural gene? Structural gene means it's a unit which get transcribed and translated. It undergoes transcription, it makes an RNA, it undergoes translation, it makes a protein, then that is called as a structural gene, right? Then, what do you mean by constitutive genes? Another word came. Constitutive genes, children. Constitutive genes are also called as housekeeping genes, I am telling. Constitutive genes are also called as housekeeping genes. Now, what do you mean by housekeeping genes or constitutive genes? Means, see, a housekeeper, so they don't have Sundays, Mondays, they don't have holidays, every day they have to come, they have to maintain the house, right? So like that, housekeeping genes means these genes are always expressed in the cell. The genes which are always expressed in the cell, right, are called housekeeping genes. Housekeeping genes are also called constitutive genes. What are they? So they are the genes which are always expressed. Now, to help you understand little more better, I will tell you. Glycolysis is a process of breaking glucose. Why do we eat glucose? Why do we take food? So that when you break glucose, you will get energy. Now, the breakdown process of glucose is called glycolysis. We know that glycolysis is a 10-step reaction mediated by 10 enzymes, right? So, all the enzymes we know they are proteins. That means they are synthesized by transcription, translation. So, that means the genes are coding for glycolytic enzymes and the enzymes are breaking glucose. Then you are getting energy. With that energy, you are doing your work. I am doing my work. So, that means glycolytic enzymes, do they work continuously then? Are they called as constitutive genes then? Yes. Understood? So, can I write down here? The glycolytic genes are examples of constitutive genes because glycolysis every second in your body should happen. Then only you will get energy. With that energy, your body is functioning. So, if you want to see examples for constitutive genes, take glycolytic genes. These glycolytic genes will code for glycolytic enzymes. These enzymes will give us energy by breaking glucose. So, now you understood what are constitutive genes. Now, let us see the other way also. Non-constitutive genes also we will see. The next one is non-constitutive genes. Now, if you are telling that constitutive genes are housekeeping genes, they are always expressed, then you can tell that non-constitutive genes means they are luxury genes. So, what's the other name? They are called as luxury genes. Now, what do you mean by luxury genes? Luxury genes means they are expressed only when there is a requirement by the cell. So, this one they are expressed always, whereas luxury genes means genes which are expressed. When are they expressed? Only when there is a requirement. Only when there is a requirement. The genes which are getting expressed only when there is a requirement, that means they are non-constitutive genes, they are called as luxury genes. Now I am going to explain you Lac Operon. In the Lac Operon, in the next what is that board, I will show you the Lac Operon picture also. Just I will write down here, like the Lac Operon genes, the Lac Operon structural genes are examples of luxury genes or constitutive genes. How it is, when I teach you, then you will understand better. Okay. So just now let us take down an example. When you are understanding the concept of Lac Operon, then it will be very much clear for you. Now the examples of luxury genes or non-constitutive genes, I told I'll take the Lac Operon genes as examples. Means I'm just taking Lac Z, Lac Y and Lac A as examples of non-constitutive genes or luxury genes. Okay. Now we understood what is structural gene. Structural gene means which gets transcribed. Constitutive gene means which are always expressed. Non-constitutive genes means which are expressed only when there is a requirement. There is another concept called pseudogenes, you know. There is another concept is also there. What is that? Pseudogenes. Then what are pseudogenes? Pseudogenes are genes, but during the course of time they have forgot or they have lost their function. They are unable to code for a protein. Then they are called pseudogenes. What are pseudogenes? They are false genes. Pseudo means false. They are false gene during a course of time. So they lost their function. So pseudogenes means genes which have lost their function. They are not able to code for the proteins. Earlier they were able to code, but now they have lost the ability to code for the proteins. Then they are called pseudogenes. So I think with this introduction, we can step into the concept of Lac Operon, right? So this is the introduction. So I told you our today's topic is 6.8.1, the Lac Operon. Then to take you to Operon, I explained few more words here. Like Benzer is a scientist who coined the term cistron, muton and recon. Cistron means a unit of a gene which codes for one protein. Recon means unit of a gene which undergoes recombination. Muton means a unit of a gene which undergoes mutation, right? Now, in the same manner, Jacob and Monod came, they used another word called operon. What is operon? Gene which is under regulation, right? So, whatever board you are, in whatever board you are studying, that board is regulating you. This is your precise syllabus and this is your exam schedule. So, they are regulating you. Like that, the gene is also which is under regulation to be called as operon. Now, who explained the concept? You are telling that operon is coined by these and these scientists. They explained the Operon model also. Which year, ma? 1961. What are the names of the scientists? Francis Jacob, who is a geneticist, with Jacques Monod, who is a biochemist. They have proposed the Operon model to explain the concept of regulation in prokaryotes. Now, we have seen few names because in the content it comes constitutive genes, it comes non-constitutive genes, it comes. Structural gene means which undergoes transcription, which makes RNA, which further makes protein. Non-constitutive genes means which are expressed only when there is a requirement. Constitutive genes means which are always expressed. Pseudogenes means genes which have lost their function during the course of time. Take a screenshot if you want. Then we will get into the topic of Lac Operon. Right? Now let us see the concept of Lac Operon. Yes. So we tell that in prokaryotes, regulation is at only one level, that is transcriptional level. So in prokaryotes, in prokaryotes, regulation of gene expression. Regulation of gene expression. If you want to regulate the gene, so you have to regulate only at transcriptional initiation. It is only at one point, that is transcriptional initiation. We talked it in the previous lecture, right? So in prokaryotes, regulation of gene expression is only at one point, that is transcriptional initiation. Now the Jacob and Monod tried to explain prokaryotic regulation. How can you control the transcriptional initiation? By taking Operon as an example. So Operon, so in 1961, Jacob and Monod, they coined the terms operator. They used the term operator. They used the term Operon also. Till then these words are not there. They introduced the term operator and they introduced the term Operon. So here we will take Lac Operon as an example. Or when we talk about operons, basically you can classify them into two types of operons. One is called as inducible operon. The second one is called as repressible operon. What's the first type? Inducible operon. And what's the second type? Repressible operon. Okay, children, there are two types of operons. Inducible operon is the first type of operon and repressible operon is the second type of operon. For inducible operon, we can take Lac Operon as an example. What's an example for inducible operon? We will explain with Lac Operon. What's an example for a repressible operon? Tryptophan Operon. Example is Tryptophan Operon. Right? In Lac Operon, NCERT says that Lac stands for lactose. So came in the NEET previous paper also. They asked us in the NEET previous question, what is the meaning of Lac in Lac Operon? It stands for lactose. What is lactose, children? It is a disaccharide. It is a disaccharide. Now, in Tryptophan Operon, so what will TRP stands for? TRP stands for an aromatic amino acid called tryptophan. TRP stands for tryptophan. What is tryptophan? It is an aromatic amino acid, right? So let us try to understand what is inducible operon and what is repressible operon. So for inducible operon, we are taking Lac Operon as an example. For repressible operon, we are taking Tryptophan Operon example, and you might be having doubt, what is Lac and what is tryptophan? So I am telling Lac stands for lactose, which is a sugar disaccharide. Tryptophan stands for, TRP stands for tryptophan, which is an aromatic amino acid. Now here in inducible operon, so the repressor is active. In inducible operon, the repressor will be active. What is repressor? You will understand. The repressor is active, and we can tell normally in inducible operons, repressors are active. But in the presence of inducer, if inducer is not there, then the repressor protein is active, children. But in the presence of inducer, repressor becomes inactive. In the presence of inducer, repressor becomes inactive. That is the case in inducible operon. Whereas in repressible operon, we will tell that normally the repressor is inactive. Normally, the repressor is inactive. The repressor is inactive, and in presence of inducer, it is inactive. in the presence of inducer, it becomes active. In presence of inducer, it becomes active. So, we'll take Lac Operon, then we'll get better clarity. We are dividing operons into two types called inducible operons and repressible operons. Example for inducible operon is Lac Operon. Example for repressible operon is Tryptophan Operon. In inducible operon, Lac stands for lactose. In Tryptophan Operon, TRP stands for tryptophan. Lactose is a sugar, tryptophan is an amino acid. So, in inducible operon, we are telling that the repressor protein will be active here, but there the repressor protein is inactive there. Okay. Then in inducible operon, so it can also be taken as example of catabolic operon. It can be taken as an example of catabolic operon because what happens in inducible operon is the lactose gets catabolized, the disaccharide is broken. That's why we can call it as a catabolic operon. Whereas in repressible operon, we can call it as an anabolic operon. We can call it as an anabolic operon. Means in tryptophan operon, the genes will code for the synthesis of tryptophan. The genes will code for the synthesis of tryptophan. Okay. So if you want to see once again, let's take the picture. Here I am drawing a Lac Operon and here I need to draw the Tryptophan Operon. In Lac Operon children, there are three structural genes. So one gene is called Z, another gene is called Y, the third one is called A. So in Lac Operon, the structural gene has three parts: Lac Z, Lac Y, Lac A. All these three genes, they will code for enzymes. These enzymes will help in breakdown of lactose into monosaccharides called galactose and glucose. So, since the enzymes which are present in the Lac Operon ZYA are making the enzymes and the enzymes are useful for the breakdown of lactose disaccharide into two monosaccharides, we are telling Lac Operon, inducible operon is an example of catabolic operon. Whereas if you take anabolic operon, in anabolic operon structural gene, there are 5 parts. So 1, 2, 3, 4, 5. So we'll tell tryptophan A, tryptophan B, tryptophan C, tryptophan D, tryptophan E. So here ZYA only, but they are A, B, C, D, E from that order. Okay. All these 5 genes will code for 5 enzymes. All the 5 enzymes are required for synthesis of tryptophan. Here the enzymes are required to break down lactose, but here the enzymes are required for the synthesis of tryptophan. Since the genes are coding for the enzymes and the enzymes are helping in synthesis of tryptophan, we are taking it as anabolic operon. Hope you understood the concept. We are dividing the operons into inducible, repressible. In inducible, Lac Operon is an example. Lac Operon, we'll see it in detail, right? So, for repressible operon, Tryptophan Operon is an example. In Tryptophan Operon, the structural gene has 5 parts. All the 5 parts will code for 5 different enzymes. All the enzymes will help to synthesize tryptophan, right? If you want to take a screenshot, then we will get into the concept of Lac Operon. Yeah, come on. Now, shall we draw the picture of Lac Operon and neatly, and we'll try to understand the concept of Lac Operon which is there in NCERT book. I'm telling that an operon has 2 parts. First part is a regulatory gene and the second part is the structural gene. So, an operon has 2 parts. An operon, I'm drawing Lac Operon now. It has 2 parts. Half of it is called regulatory gene. Half of it is called structural gene. So, this side is regulatory gene. This is called a regulatory gene. And this is called structural gene. This is called structural gene. Just now we saw that the structural gene has three parts, Lac Z, Lac Y, Lac A. Let us keep it there. It has three parts, Lac Z, Lac Y, Lac A. The structural gene of Lac Operon has three parts, Lac Z gene, Lac Y, Lac A. Whereas the regulatory gene of Lac Operon also has three parts. Okay. So we call it as operator gene, promoter gene, inhibitor gene. So it also has three parts, operator, promoter, inhibitor gene. Okay. And this inhibitor's promoter will be here. So this actually, in previous lecture we discussed that each operon has one promoter and one operator. Each operon will have one promoter and one operator. That means for this operon, this is a promoter and this is an operator. So don't get confused looking at this promoter. Oh, madam has got two promoters. No, this is the promoter for the I gene but not for the structural gene, right? For this structural gene, this is a promoter and this is an operator. Okay. Now let us see. What did I tell about I gene? I told I stands for inhibitor. I stands for inhibitor gene, or you can call it as a repressible gene. We can call it as a repressible gene, right? Why are we calling it as a repressible gene? And one more thing, the I gene, the repressible gene, the regulatory gene in Lac Operon comes under constitutive gene. So the I gene, what it is? I am telling it is constitutive gene. I told you what is constitutive gene. Constitutive gene means housekeeping gene. No rest for it. It has to continuously express. That means this I gene will undergo transcription then. The I gene will undergo transcription then. So what did it has undergone here? Transcription. Children, it will undergo transcription and it has making a RNA. This is called repressor RNA. Repressor gene, what it will make? Repressor RNA. So what is this called? It is called repressor RNA, mRNA. You also know that the RNA undergoes translation to make a protein. So now let us write down here. It is translation. The repressor RNA undergoes translation to make a repressor protein. Then it will make a protein. The protein is called repressor protein. What is the protein called? The protein is called repressor protein. So the repressor RNA undergoes translation to make a repressor protein. So Lac Operon is an example for inducible operon. In inducible operon, I told you that the repressor protein is active. Did I tell you that the repressor protein is active? So keep that in mind. The repressor protein is active then. The repressor protein is active. Yes, it is active. So the repressor protein is active and the repressor protein has affinity for the operator. Like among biology, physics and chemistry, you have affinity for biology because it is easy, it is scoring, right? So in the same manner, the repressor protein has attraction. It has affinity towards operator. Okay now? So this repressor protein, it has attraction or it has affinity towards what? It has affinity towards operator, I am telling. It has attraction towards operator. Repressor is having attraction towards operator. Since it is having attraction towards operator, children, the repressor protein will go in search of operator. Here it is, it found the operator. It will go and stick there. So the repressor protein goes and binds to the operator. See, the repressor protein goes and binds to the operator. Okay now. So the I gene, inhibitor gene, which is a constitutive gene, which will always express. So it has undergone transcription and it has made a repressor RNA. The repressor RNA undergoes translation, makes a repressor protein. The repressor protein is active. Since it is active, it shows its attraction towards the operator, searches the operator, goes and binds to the operator, blocks the operator. When the repressor protein goes and binds to the operator, we can tell that in this situation, operator is blocked. In this position, the operator is blocked by the repressor protein. When the operator is blocked, then, so now we'll see. We understood about I gene, everything in detail. Okay. Now let me talk about P gene also. Let me talk about P gene. We finished about I gene. Now let's talk about P gene. What is P? P stands for promoter. I told you. P stands for promoter gene. And I think we know that in a transcription unit, promoter, structural gene, terminator will be there. Promoter is the place where RNA polymerase identifies and comes and binds and starts the process of transcription, isn't it? Now, what is the function of promoter gene? Promoter gene is identified. Promoter gene is identified by whom? It is identified by the RNA polymerase. Is identified by the RNA polymerase. So, it wants to come and bind here to the promoter gene. When if it comes and binds here, then it wants to move on, it wants to slide on, because where are the structural genes from here? LAC-Z, LAC-Y, LAC-A structural genes. Now, if the promoter comes and binds, RNA polymerase enzyme comes and binds to the promoter, come on, tell me, can it move on the operator? Is the operator free or the operator is blocked? Operator is blocked. So, then RNA polymerase cannot come and bind to the promoter. When RNA polymerase cannot come and bind to the promoter, then will the structural gene transcription happen? No, it will not happen. So, the function of promoter gene is, it gets identified by the RNA polymerase. It gets identified by the RNA polymerase, and this RNA polymerase will do the transcription. It will do the transcription of the structural gene. It will do the transcription of the structural gene. Right? So, now you understood very much about the promoter. Now, let us discuss about operator also then. What is O? I told you, Jacob and Monod are the scientists who coined the word operator, who coined the word operon also. Now, they are the people who introduced the concept of operator. O stands for operator gene. O stands for operator gene. Now, just think and tell me, is it the operator who is deciding? Is it the operator who is deciding whether the structural genes should be transcribed or not to be transcribed? Yes. If the operator gene is free, then only the RNA polymerase will bind, then only the RNA polymerase can move, then only the RNA polymerase can read, then only it can do the transcription and translation, then only you will get the proteins, correct? Now, in this case, the operator is blocked. When the operator is blocked, then the RNA polymerase cannot bind, it cannot move, it cannot read, it cannot do the transcription. So, what is the role of operator gene? Operator gene decides whether the structural gene should be transcribed or not transcribed, right? Operator gene, it decides whether the structural gene, is to be transcribed, it is to be transcribed or not. Whether it has to be transcribed or not, who will decide? Operator will decide. So, I think I explained what is the role of I, what is the role of P, what is the role of O. What is the role of I? I is constitutive genes. It always produces, it's always constitutive, it always gets expressed, makes a repressor protein. The repressor protein is active in the case of Lac Operon. It searches for operator, goes and binds to the operator. This role finished. Coming to promoter, the role of promoter is to be identified by the RNA polymerase. RNA polymerase should come and bind, start the process. Finished. Operator. The role of the operator is to decide whether the transcription should promote structural gene should be done or should not be done. If the operator is free, then transcription will be done. If the operator is blocked here, if the operator is blocked, then the transcription is not done. Understood? Now, let's talk about the structural genes also then. Let's talk about, we finished about the regulatory gene. The regulatory genes are 3, I, P, O. The structural genes are also 3 for Lac Operon, Z, Y, A. Now let's talk about what is Lac Z, what is Lac Y, and what is Lac A. Lac Z gene. Lac Z gene is the first structural gene. It's the first structural gene. Now Lac Z gene codes for an enzyme called beta galactosidase. So what does Lac Z codes? Lac Z codes for an enzyme. It codes for an enzyme. What is the name of the enzyme? Beta galactosidase. The name of the enzyme is beta galactosidase. This Lac Z gene will code for an enzyme called beta galactosidase. Now, this beta galactosidase has two functions. It has two functions. One function we already know. It breaks the glycosidic bond. In lactose is a disaccharide made up of two monosaccharides. This is lactose. It's made up of two monosaccharides. One is glucose, other one is galactose. Both are linked by glycosidic bond. Now, if you bring this enzyme, beta galactosidase, it breaks the bond between them. They get separate. You will get two monosaccharides. So, the first function of this beta galactosidase enzyme is to break the lactose. To break the lactose into glucose and galactose. It breaks the lactose into glucose and galactose. That's the first function. Okay, now. Then coming to the second function. What is the second function of beta galactosidase? So, it can also convert lactose. It can also convert lactose into its isomeric form called L-lactose. Into its isomer called L-lactose. This is the second role. This is the second role of beta galactosidase enzyme. Beta galactosidase enzyme has two roles to do. One role to break the glycosidic bond in the disaccharide and make the monosaccharides out. The second function is to break the lactose into, to convert the lactose into its isomer called L-lactose. So that is about Lac Z. Now let's talk about the second one. It is Lac Y. What is the role of Lac Y gene? What is the role of Lac Y gene? Lac Y also codes for an enzyme. The name of the enzyme is called permease. What is the name of the enzyme? Permease. This permease enzyme which is present in the cytoplasmic membrane. The enzyme which is present in the cytoplasmic membrane. Now what it will do? The enzyme which is present in the cytoplasmic membrane. If you want to enter into a college, watchman should permit you inside the campus premises. In the same manner, if the lactose which is there outside in the medium, if it has to come into the bacteria, means the cytoplasmic membrane, the cell membrane should permit inside, right? So the permease is like a watchman. It is on the plasma membrane. It permits the lactose to enter into the cell. So what is the permease doing? Permease will permit. It will permit the entry of what? Lactose. Permease will permit the entry of lactose into the cell. Yes, it is into the cell, children. So I told the function of Lac Z. I told the function of Lac Y also. The third gene is called Lac A gene. What is the third gene called? The third gene is called Lac A gene. Now Lac A, then we understood they are coding for enzymes. What enzyme madam is going to tell now? Lac Z, beta galactosidase. Lac Y, permease. Lac A is coding for an enzyme called trans-acetylase. It codes for an enzyme called trans-acetylase. Now NCERT, 12th NCERT says that the function of trans-acetylase is poorly understood. The function of trans-acetylase is poorly understood, it says. But if you go to higher end textbook, they will tell that it helps in acetylation of monosaccharides. So trans-acetylase helps in adding acetyl coenzyme A group. It helps in acetylation of monosaccharides. Higher end textbooks say that it helps in acetylation of monosaccharides. It helps in acetylation of the monosaccharides, adding acetyl group to the monosaccharides. It will do. But NCERT says that the function is not clearly understood, it is poorly understood. So now I talked about all the three genes which are under regulatory genes: I gene, P gene, O gene. I gene we discussed here, P gene we discussed here, O gene we discussed here. And then Lac Operon has three structural genes: Z, Y, A. Z gene, two roles we discussed. Y gene, one enzyme we discussed. A gene function is not clearly understood. So in this order you have to maintain, right? So you understood till here. Now we are telling that this operon contains the genes which can be regulated. Which can be regulated means when there is a requirement, then only they should get expressed. Then only this enzyme should be there. Okay, let us see what is that concept. Now take a beaker. Now in this beaker you are having lactose medium. In the beaker you are having lactose medium. Now in this lactose medium you have kept a bacteria, E. coli. Now these are the bacterial colonies. These are the bacterial colonies which are inside the medium. Now that means the environment which is around the lactose, which is around the E. coli is lactose. So this is the bacteria. It is in an environment where lactose is there. That means it should have a mechanism to take the lactose from there to get the energy, right? So then when the lactose is present in the medium, what will happen? Now let's just discuss. When the lactose is present in the medium, what happens? We will discuss, right? So the lactose which is present here, first it will enter inside. The lactose which is present in the surrounding medium will enter into the bacterial cell. It will enter into the bacterial cell. Now, what is lactose? We need to discuss now. We need to discuss what is lactose also, right? Now, I am telling that lactose is an inducer. That is why we told Lac Operon is an inducible operon. Here, the lactose is acting like an inducer, inducing something. I am inducing the subject into your brain, right? So, like that, lactose acts as an inducer. Now, lactose acts as an inducer. The inducer came inside. When the inducer comes inside, what it will do? So, this inducer particle, lactose, has affinity. Lactose has affinity for repressor. Lactose has affinity for which protein? Repressor protein. Children, the lactose came inside. So, when your grandparents comes, they will be finding like, where are the children? Where are grandchildren? Because they have more love and affection towards the grandchildren. In the same manner, lactose came inside. Now, lactose has attraction for repressor. It is searching inside the cell. Where is repressor? Where is repressor? It will find that the repressor is here. The lactose will find the repressor is attached here. Now, this lactose which acts as an inducer. The lactose which has an inducer, which acts like an inducer, which has affinity for the repressor, will go and bind to the repressor. It will go and bind to the repressor. The inducer which has attraction for repressor goes and binds to the repressor. It goes and binds to the repressor, and when the lactose goes and binds here, this is lactose. So, when the inducer goes and binds to the repressor, you know what happens? There will be a change in the conformation of the repressor. So, when lactose comes and binds to the repressor, then what happens? I am telling, then there is a change. There is a change in the conformation. There is a change in the conformation. What do you mean by conformation? Structure, I am telling. There is a change in the structure of which protein? Of repressor protein. There is a change in the structure of the repressor protein. When inducer goes and binds to the repressor protein changes, it seems. When it changes, then structure is changed, then it can no longer bind to operator. Changed repressor now becomes inactive repressor. Changed repressor becomes now inactive repressor. Changed repressor becomes inactive protein. It becomes inactive repressor. Children, when it is inactive, can it bind here? It cannot bind. It will come down. The repressor protein comes down. It drops down. When the repressor protein drops down, then the operator is not blocked, operator is free. When the operator is free, then the RNA polymerase can bind here. When the operator is free, this is the RNA polymerase enzyme, sigma factor. The RNA polymerase will come and bind here. When the RNA polymerase comes and binds here, it will slide. When the RNA polymerase comes and binds here, it will slide. Finally, it has reached the structural gene. When it has reached the structural gene, then what it will do? Fast, it will do the transcription. It will do the transcription of the structural gene. This is a structural gene I told. It will do the transcription of the structural gene. So transcription means making RNA. Then you will get an RNA. So this RNA, which is coding for 3 proteins, right? One protein will be beta-galactosidase, another protein will be permease, other protein will be transacetylase. So one RNA is coding for 3 proteins. So can we call it as a polycystonic RNA? In prokaryotes, P with P. In prokaryotes, it is polycystonic RNA. So in the presence of inducer, I am recapping it. In the presence of inducer, inducer has attraction for repressor. It will go in search of repressor. It locates the repressor, binds to the repressor, makes the repressor inactive, changes the structure of repressor. Then the repressor structure is changed. It becomes inactive. It can no longer stick there. It falls down. When it falls down, operator is free. When the operator is free, the RNA polymerase comes and binds to the promoter. It moves, it reads the sequence, and it will make a polysystonic mRNA. It will make a polysystonic mRNA. Why are we calling it as polysystonic mRNA? Because one mRNA will make many proteins, right? Now this polysystonic mRNA undergoes translation. It undergoes translation. When it undergoes translation, it will make one enzyme, second enzyme, third enzyme. So the first enzyme is this, right? The first enzyme coded by Lac Z is beta-galactosidase. So this is enzyme 1. And it codes for second enzyme, Lac Y. Lac Y codes for permease. This is enzyme 2. The third one is Lac A. Lac A codes for transacetylase. This is enzyme 3. So what it will do? It will undergo translation and it will produce all the 3 enzymes, beta-galactosidase, galactoside permease and transacetylase enzymes. Now these enzymes, what they will do? Now these enzymes will come and they'll break the lactose. They'll come and they'll break the lactose. When you break the lactose, you get glucose and galactose. Now when you are breaking anything, you get energy. That means the cell is getting energy. With that energy, the cell will survive. The cell will be alive, right? So in this manner, see how much complex the process is. That's why we told regulation is not a simple term. It's a broad way of explaining it. So then the enzymes are made. The enzyme will break the lactose. Then you will get energy. So when this concept is called operon concept. When, if I ask you, when is the operon switched on? When is operon switched on? When are the structural gene transcribed in presence of lactose? Yes. So take a screenshot of this. I'll explain you writing the sentences on the board. So we are doing Lac Operon. I draw the Lac Operon's gene here. I told Lac Operon gene has 2 parts. Half is the regulatory part, half is the structural part. I showed you this is the regulatory part, this is the structural part. Its regulatory part has 3 genes called I gene, P gene, O gene. I gene is an inhibitor gene. I is also called repressible gene. I is called constitutive gene. It's always expressed what it will code for. It will code for a repressor protein. What is the role of repressor? Repressor has attraction for operator. It goes and binds to the operator, blocks the operator. Promoter, we know RNA polymerase identifies the promoter and goes and binds there. That's the role of promoter. What's the role of operator? Operator is the one which is deciding if the transcription should happen, operator should be free. If the transcription should stop, operator should be blocked. Now structural gene, Lac Z, Lac Y, Lac A. Lac Z codes for an enzyme, beta-galactosidase. Beta-galactosidase has 2 roles to do. One, to break the lactose into monosaccharides, glucose and galactose. The second role of beta-galactosidase is to convert the lactose into its isomer called yellow-lactose. Lactose and yellow-lactose. Lactose and yellow-lactose are inducers. They are inducers. We understood what is an inducer. Inducer has affinity for repressor. Inducer goes in search of repressor, goes and binds to the repressor and it pulls the repressor down. When the repressor is down, operator is free. When the operator is free, RNA polymerase binds. When the RNA polymerase binds, then the structural genes are read. Transcription happens. mRNA is made and undergoes translation. It makes 3 proteins, Lac Z, Lac Y, Lac A. This enzyme will help to break the lactose to get energy. This enzyme will permit another lactose inside. The third enzyme function is poorly understood. Take the screenshot fast. Right. So now you understood. So now let us write down it in a better manner. diagram I explained. Now let us write on. Now we will understand it clearly once again. The control of Lac Operon, right? So we will talk in two terms. When inducer is present, what will happen? When inducer is absent, what will happen? So that's why I wrote two times. So here we'll take a condition. What is our condition? What is our condition? We are telling the condition is in absence of inducer. Our condition is in absence of inducer. This is our condition. Now, the next one is in presence of inducer, what will happen? I'll erase this also. And we are telling that, what is this condition? This condition is in presence of inducer. The condition here is, what happens to the operon? In presence of inducer. And here we are telling, what is the condition we have kept here? In absence of inducer. Now children, you understood, I think, who is inducer? Lactose is inducer. Who is inducer? Either lactose or yellow lactose. Either lactose or yellow lactose is a inducer. Now let us talk. In absence of inducer, what will happen? If inducer is not there. If inducer is not there, then the I gene undergoes transcription, makes a repressor RNA. The I gene, what does it undergo? It undergoes transcription. It undergoes transcription and it makes a repressor RNA. Now this undergoes translation. This undergoes translation and it makes a repressor protein. It makes a repressor protein. And as I told you, in Lac Operon, the repressor protein is active. And I am telling you, the repressor protein has affinity for, the repressor protein has affinity for operator. As soon as it is made, it is searching where is operator, where is operator. It finds the operator here. As soon as it finds, its search got over, it will go and bind to the operator. As soon as it finds, it goes and binds to the operator. Now come on, tell me what happened to the operator. Operator is blocked. What happened to the operator? Operator is blocked. Children, when the operator is blocked, then RNA polymerase enzyme cannot come and bind here. When RNA polymerase enzyme cannot come and bind here, it can't read the structural gene. When the structural gene is not read, that means there is no transcription. So in this case, we can write that no transcription. Structural genes are not transcribed. No enzymes are made. No enzyme production also. Enzyme is not made, transcription is not done. When is this? In the absence of lactose. In the absence of lactose. See, if the structural genes are not transcribed, if the enzymes are not made. So, is it apt to use the word here that the operon is switched off? It's not doing anything. So, can I tell this is a condition where we can tell that the operon is switched off? Yes. So, can we tell that the operon is switched off? Yes, it is off. Then now come on, when is the operon switched off? It is in the absence of the inducer. In the absence of the inducer. Who is inducer? Lactose is an inducer. Inducer is there or not there? Your condition is lactose is absent. When lactose is absent, do you need the enzyme beta galactosidase to break it? Do you need the enzyme to permit it? No one is there outside. Whom will you permit? No one is there. Whom will you break? So then, unnecessarily energy should not be wasted. That means it's better that the operon is switched off. It is conserving its energy. Understood? Now let's talk about the other condition. What happens in presence of inducer? What happens in presence of inducer? So it will be, the first method is the same, that the I gene makes an RNA and RNA makes a repressor protein. Repressor protein goes and binds to the operator. Repressor protein goes and binds to the operator, right? Till here it is same. Now next I am telling, if inducer is there, who is inducer? Lactose or allolactose. If lactose is there, what will lactose do? So lactose, I am telling, lactose acts like an inducer. Lactose acts like an inducer. It is inducing, it acts like an inducer, and inducer has affinity for, inducer has attraction for what? Inducer has affinity for repressor. Inducer has attraction for repressor, then obviously it will go in search of repressor. Here is a repressor. When inducer sees the repressor, the inducer goes and binds to the repressor. The inducer goes and binds to the repressor. So then what happens? When the inducer binds to repressor, children, tell me what happens. The repressor protein's conformation got changed. When inducer binds to the repressor becomes inactive. It becomes inactive. Repressor protein changes its conformation. It changes its conformation. Amma, when it changes its conformation, then it can no longer bind to operator. So repressor can no longer bind to operator. It can no longer bind to operator. Then if it can no longer bind to operator, then can I tell that this came out? You are telling that it became inactive, it can no longer bind to operator. Then operator becomes free. And what happened to this repressor? Its conformation changed because it is with inducer. It came out and it became inactive. Now in such a condition, operator becomes free. See there, operator is free. So when this happens, we are telling that operator becomes free. When the repressor falls down, operator becomes free. When the operator becomes free, then here the RNA polymerase will bind. RNA polymerase can move. It can read the structural genes, right? RNA polymerase enzyme locates the promoter, sigma factor, goes and binds there. It moves on the structural gene. So RNA polymerase, what it will do? It will move on the structural gene and it will do the transcription of a polycystonic mRNA. It will do the transcription of polycystonic RNA. This polycystonic RNA will code for first enzyme, second enzyme, third enzyme. Will code for first enzyme, second enzyme, third enzyme. The first enzyme, so Lac Z, right? Lac Z codes for an enzyme called beta galactosidase. The Lac Z codes for an enzyme called beta galactosidase. When is beta galactosidase required? When lactose is there. Is lactose there? Yes, it is there. In the presence of inducer, you are telling. Inducer means lactose only. So if lactose is there, then you need the enzyme to break the lactose. Yes. So then the Lac Z produces beta galactosidase. This beta galactosidase breaks the lactose disaccharide into two monosaccharides called glucose is one, other one is galactose. Right? And then the second enzyme is coded by Lac Y. Lac Y codes for permease. And what will permease do? Permease will permit the lactose molecules inside. It will permit lactose molecules inside. And what will Lac A do? Lac A codes for an enzyme called trans-acetylase. But they told the function is poorly understood. The function is poorly understood. So children, in the presence of lactose, what happened? All the enzymes are made. If all the enzymes are made, what do you say? The operon is switched on, operon is switched off. It is switched on, right? So in this condition, we can tell that the operon is switched on. Very good. The operon is switched on. When is the operon switched on? Operon is switched on in the presence of the inducer when there is a requirement. And when there is no inducer here, then the operon is switched off. Here, unnecessarily, why you should waste energy when the enzymes are not required? It's switched off. When there is no one in the room, we'll switch off all the lights and fans. When there is an inducer, you need the enzymes to break it. Then the operon is switched on. So see how nicely the gene is regulated. So Lac Operon can be taken as an example for positive regulation also. It can be taken as an example of negative regulation also. So what is positive regulation? Positive regulation means it is with the repressor. Negative regulation means it's with the repressor protein. So negative regulation means when the repressor protein is active. Amma, tell me, when the repressor protein is active. Here I told repressor protein is active. When it is active? In the absence of inducer. So repressor protein is active when in absence of inducer. In absence of inducer. In absence of inducer, where the repressor protein is active, then we tell that the operon is switched off. Then the operon is switched. That's what we wrote. It is switched off. Now this regulation where it is stopped is called as negative regulation. Now what is positive regulation? In NCERT they tell that positive regulation is beyond the scope of the textbook. Positive regulation is beyond the scope of the textbook, but then we can tell that there is a protein called CAP protein, called Catabolic Activator protein. What is that? Catabolic Activator protein. CAP protein gets activated by cyclic AMP. CAP protein gets activated by cyclic AMP. So when cyclic AMP is there, it activates CAP. When CAP is there, then the operon is switched on. When CAP is there, then the operon is switched on. So when the operon is switched on, that means it is making the enzymes. Then we tell it is positive regulation. So Lac Operon can be taken as an example for positive regulation also, negative regulation also. Positive regulation means in presence of CAP and cyclic AMP. Negative regulation means so in absence of inducer, where the repressor is active, that comes under negative regulation. So you understood clearly about the operon concept. Any queries, you can post it in the comment box. So in this session, we discussed in detail about Lac Operon and its regulation. Positive regulation, negative regulation, in absence of inducer, in presence of inducer. You can take the screenshot. And in the next topic, in the next lecture, we'll talk about Human Genome Project. Hope you like the lectures. If you like the content, like, share and subscribe to my channel. Thank you.